All right, thanks everybody for having me. I work at ONDA, which is a contract research organization here in PEI, and I actually wasn't sure how familiar the crowd would be with ONDA, so I did a little intro to us at the start. But we have two spots, one in Surrey, one in Victoria, over 400 different tank systems, 22 rooms that can be fully biocontained, and we work with species all over the world. Certainly the client base is very global. And until MSX and Dermo, we didn't have very much interaction actually with any of the Eastern oyster industry. They're very new to us. We work with a lot of species, like I mentioned. So Atlantic salmon would be about 70% of our business. Rainbow trout would follow behind that. And then we have a swath of different species afterwards that we work with. And so oysters definitely have experience with it, lots of breeding programs, but it's always been Pacific oysters for us. So we've worked in the States, British Columbia, and Mexico up until last year. And on the team, we have 65 team members in total. 10 of them are PhDs, 16 MSCs, and 3 vets on the team. We publish a lot, so we do our own academic bit, but the main priority, of course, is actually for our contract customers. So most of our work actually doesn't see publication. And we have a wide variety as well of nationalities that work there. It gives us a good diversity and expertise. But maybe one interesting thing to say is that none of us are oyster experts either. So until this kind of came about, there wasn't a large intention toward oysters for ONDA. So I come from the lab technologies group. We'll just give a quick overview of that, and then I'll get into some of the work that we've done on the parasites. We do custom qPCR build-outs for labs around the world. So we sell the validations. I think what Rebecca just talked about is a clinical diagnostic validation, which is not what ONDA is doing. And so I'll explain the difference between how the two tests are validated as well in the talk. We do a lot of different functional screening as well, gene expression, microbiome, and then we screen treatments. This is actually the bread and butter of the company. And so different vaccines, different alternative treatments, and there are some for oysters, some cool inventive ideas. I won't be able to talk about a lot of those, obviously, because they're private for now. For the oyster industry and for Eastern oysters specifically, we've tried our best to develop as many analytical services as possible based on demand. So we did develop the qPCR screening for MSX and Dermo. When I say develop, we transferred it. These are tests that have been developed by other people in the past, been validated by other labs in the past, and so we do our own validation, but you'll notice it's not an ISO 17025 validation. This is a validation rather that we use to sell tests into other labs that will then ISO it themselves. So the point I would try to make here is around our limit of detection, which I think is really what is important in the ISO 17025. There's a really rigorous testing of that limit of detection. Our validation is a little bit softer on that. And so we're really curious right now, especially in seed juveniles. We see a lot of low-lying results coming out of those, never high. 33 CQs, I think, is the lowest that we've seen. What does that mean? We're not exactly sure yet, for example. So still learning with the qPCR test. But we've done over 13,000 oysters this year, so we've been busy. Like we're not oyster experts, we're also not a diagnostic lab by trade. So it was definitely a step up for us to try and service all of that. And we've done over 30 different sample types testing for MSX as well, which I think will be of interest in the next couple slides. We do some functional support on the oyster, so what happens when they get disease? What is their response to it? Do they have an immune response, and if so, what does it look like? And then what bacteria accompany the two parasites? So we look at the community within the oysters, within the water. How does that change? Is there any predictive metrics within those bacteria that we can use to help screen for the disease? Then, like I mentioned, treatment systems. We're doing a lot of different angles for the treatment of MSX and Dermo right now. Water quality analyses, parasite bioassays, and then disinfection type assays as well. We're currently working on another way to detect DNA. That's what qPCR does. It detects the DNA from these parasites. Doesn't necessarily mean that you have a viable infection, of course, and I think we have a histology talk after that, so I'm guessing it'll be mentioned. But qPCR is a good initial screening tool. And so we wanted to be able to do it a little bit more rapidly. We noticed that we're bottlenecked quite badly by the processing of the oysters themselves. This takes a lot longer than most sample types that we're used to receiving, admittedly. And so having a test that farmers could do on their own, whether it be in an office, whether it's on the boat, very accessible, 20 minutes. And we're using a different technology. So this is pretty cool because typically these tests require an incubation at 65 degrees Celsius. And so we're trialing, a fairly new technology as far as a probe goes, where you don't need that. It'll just sit at room temperature for 20 minutes, and it'll have either a color change, which is not my preference, it's not overly sensitive, or a fluorescent change. You can read it with a UV light in the back, or this $2,000 machine that you see on the screen. So there's a few different options in the way that you could read out a test, but cheaper than the 70 grand that you'd spend on a qPCR for sure. We do a lot of different sample collection types, and this is more of our job. Like I said, we're not clinical diagnosticians, we're more surveillance is what we do, and so we take different types of samples from the bays and from rivers, trying to understand the secondary host bit as well. We're searching for it, and on to searching for that host on its own dollar, so it's not funded actually at the moment. Taking sediment from below cages and just randomly. We use FTA cards for easier shipping. It works quite well, and we validated that, and so you can homogenize an oyster and put it on one of those cards and ship it in the mail. It's a little bit easier to do. eDNA from water, tissue stabilization, which I think everybody's quite used to at this point. Water sampling, wastewater is just larger volume sampling, and then swabbing as well. And we heard a lot about the gills from Ryan's talk today, and swabs out of the gills seem to be quite promising as well. Again, on surveillance, not for diagnostics. Okay, so I'll shift gears a little bit as well. So we have the highest level of biocontainment that you can have in the world for an aquatic facility, and that as well the largest space for that. And so there are certain projects that are on the move right now. Again, we won't go too into depth on those, but that require either the containment of different genetics from the oysters or of the pathogens themselves to work on it from different places. So just letting everyone know that obviously we're offering those services and are actively have oysters in the facility right now. So just kind of highlighting some of these challenge models, and it was talked about, we do believe that for breeding, family studies are the most direct route to get there. It would be nice if it was easier to pull off, admittedly. MSX is extremely difficult as far as its biology goes. I think derma will be a little easier when we get there. But right now we offer breeding challenges that are very successful, some of the biggest companies in the world. For OSHV1, this has been kind of our biggest success. We are one of the only labs to be able to reproduce this reliably in the lab. We have a couple different Vibrio challenge models as well. And then thermal tolerance has been huge. That's probably our best seller. And just as it sounds like, exposing hundreds of oyster families to hot water and picking the best survivors and breeding them and moving again. So we also have food safety models. This is largely in depuration or effluent validation. And you'll notice these bugs from the testing you do near harvest. And then disinfection models, you probably don't notice any of these. They're actually proxy bugs. They're just really hard to kill. So we use those both on the benchtop and in vivo to understand how the parasite might transmit away from the host. So just what I was talking about with the breeding studies, just want to show you some of the data and what that would look like. There's also a lot of discussion around tolerance and resistance, so maybe hopefully clear that up here too. But we're always looking at usually hundreds of different families at the same time that are exposed to the same difficult conditions. Some of them do well and perform well, some of them don't. And the ones that perform very well typically are top 10%. We breed again and take to the next generation and just repeat this over and over again every couple of years. And so we've definitely pushed temperature tolerance in several different populations at this point. You can see, for example, here, maybe, nope. The blue group here would have better survival, or at least longer survival to this hot temperature than the red one would, which we know confers resistance in the genetic population. OSHV1 tolerance and resistance, there's a couple of different ways that we look at it. So just the mortality of oysters from OSHV1, we see that lots of oysters in our tolerant group, the green group in the middle there, they get infected, they succumb to the disease, but just less so. Versus the resistant group, we're really trying to understand how they transmit through the water. And so we have qPCR from both the oysters and in the water, and we're looking at how they transmit. We have populations that have really low levels in oysters, but they transmit through the water, no problem. So we see lots of differences depending on the family. Right now, I know the breeding is dependent on doing field breeding, which just comes with some difficulties with confounding variables. We don't always know why an oyster died or didn't die, and we have to make assumptions that it's MSX. So I think there was a question earlier on, and going one-to-one could work well, but then we could also miss it completely and miss 2 years of progress. And so that's why typically we go after multiple different crosses. But there's, for sure, lots of people in the room that can help out with those questions too and advise on the best way to go forward if you're doing your own kind of breeding approach. So yeah, we tried to do it. We tried to bring MSX into the lab to have a reliable model that people could use. And the short story is it didn't work, but I'll tell you about it anyway. So we went out to heavily infected areas. We took as much gear as we possibly could. Obviously, oysters were a piece of that, but you know that an infected oyster doesn't pass it to a naive one. So we had to try and grab the secondary host in our sampling as well. And obviously, we don't know who it is. So the goal was to get sediment, rope, bungee, cage material, dead shells, and maybe actually I'll show you exactly what we put in there on the next slide. Just know that we also fluctuated the temperature heavily. So every 2 to 3 days, we moved from 15 to 20 and then back down to 15. I'm not sure if that was stressful enough, to be honest, but that was kind of the overall design, and we had them in there for 3 months. So we tested basically everything we could find using qPCR for MSX, lots of different animals. Snails and worms definitely come up as ideal suspects when you're talking about complex life cycles of parasites. So we went after those as well, but still haven't found it yet. The maximum number of samples in any one of these that are listed here is 5. And so it's not a heavily sampled list, but more just going after diversity, if you will. So in the yellow, we have an inconclusive result. Again, for us in a surveillance test, because we don't have that LOD, as good as I think an ISO 17025 test will have, we leave an inconclusive zone at the back, which is actually considered negative. But it would be good to have confirmatory analyses on some of these inconclusives from the ISO 17025 test for sure. The reds are very positive: eelgrass, brown seaweed, an unknown algae. We're sequencing that now. We don't know what it is. We do pick up MSX off of cages. That cage was actually buried. It was overwintered in 2024. And we picked it up and swabbed it, and really high positive. And then we find it in the water as well. So, for example, in Bedeck Bay, that was hit really hard. We didn't find it in the water after most of the mortality had occurred, but in somewhere like Botten, when we had 3% positivity in qPCR, we were finding it in the water. So seems like it's temporal based on when you're going to be able to find it in the water and when you're not. And we don't know if finding it in the water is any predictive case for whether oysters will get MSX or not either. So the timeline of what we did, we just tried to bring MSX material into the facility. That was really the goal. So we brought in naive oysters from Savage Harbor on the 9th of July. And then we had 4 different exposure times where we went out to the bay, got water, 2,000 liters of water to supply the system, the rope, snails, starfish, whatever, and put it all into the system together. So from that first collection, 50% of the oysters that we'd collected from that part were positive, so we felt pretty good about that. Water was positive, cage was positive, plants and algae were positive. We went and did a second exposure. We thought Bedeck Bay would be pretty loaded up. It wasn't. We didn't really find it there at all. And we didn't find very many oysters either. Then we went back to Botten. We got a similar result, so restocked the tanks again with that infective material. About 6 weeks in, we took digestive enzymes. The hypothesis here was that if there is a secondary host, they probably do something to MSX, whether that be in a life cycle form or whether it be some type of digestion. So we took lipases and amylases and tried to digest and kind of emulate the parasite being in that secondary host. It didn't work. I thought it was a sweet idea, but it didn't work at all. And then we did a 4th exposure, again from Botten. It seemed to be our spot, a little bit less positivity on that last one. Just a few pictures from that experience. And then, yeah, so we did different samplings after that. So 15 and 50 days after we did the exposure, we got nothing. 21 and 60 oysters sampled at each time. On the 17th of September, so 64 days after that initial infection, we did get hits. Lots of them. Everything was infected almost in the system. There was no UV on, so it just was recirculating, to be clear. But all really, really low-level positives. So it was pretty clear to us that we didn't drive disease. We didn't kill any oysters. They all survived fine. And in fact, by the time we got to day 78, they were clearing it. So we found, very, very low-lying levels of MSX at that point. We believe now, and the next thing that we're doing with this study is to try and understand when we take and get infection like we did at 64 days here, what are the environmental variables that we need to push on to tip the favor toward the parasite. So that's kind of what we're working on now in some aquaria. And yeah, great. Take any questions.
We're doing questions at the end.
Oh, perfect. I can pass it on to the next speaker.